Hey, Hammy here, coming to you today with the first part of Chapter 5. Okay, in this chapter, we're going to look at cell division. How do cells divide, why they need to divide, and kind of their life cycle that they go through. We just want to look at kind of a general overview of that here in this first video, and then in the future videos, we'll get into like the specific stages that the cell goes through in order to divide itself, divide its nucleus and its contents. Okay, first, if you remember, we talked about in our first chapter that when we talk about biology or life, one of the characteristics of life is that all things are cellular. Things are made up of cells. Okay, so if life is going to keep or giving a rise to more life, cells need to be able to divide. Okay, so up here you see normally the cell that we're starting with, we call the mother cell. And that mother cell is going to divide its DNA so it has a copy for each of the offspring or each of the new cells. And then you see here the cells pinching in. Eventually, they're going to divide into two what we call daughter cells. So mother cell gives rise to two new daughter cells. Those will grow and function in our bodies or in plants or as bacteria or protozoa. And eventually those will divide, and then eventually those will divide. And so you can kind of see what's happening here, that over time, cells just keep giving rise to more and more cells, and life continues. When we discuss cell division, one of the first things we need to do is talk about the difference between prokaryotes and eukaryotes. Now remember, prokaryotes means before a kernel, before a nucleus. They don't have a nucleus. So if you look at the typical prokaryote down here, you can see that it has ribosomes, these little yellow orangish dots, and it has just this big clump or loop of DNA kind of in the center or nucleoid region, but you notice that there is no membrane around it, okay? And then it also has these little accessory DNA that when we get into the biotech chapter, we'll talk a little bit more about plasmids, because we've kind of put them to our advantage here when it comes to bioengineering. Well, anyway, here's this bacterial cell or early prokaryote, or a type of prokaryote. Okay, the process that they go through is they copy their DNA. So the plasmids have copied themselves over here, and here you see two new loops of DNA here. The cell starts to stretch itself out. Okay, it starts to form these notches all the way. It pinches in, and it'd be like taking a balloon and putting a belt around it and just pinching till it pops into two. Okay, so then you can see you have two new daughter cells over here. Okay, this process of a bacteria splitting is what we call binary fission. Okay, splitting into two. And they can do this very rapidly. E. coli can divide about every 20 minutes. And it's interesting because it actually takes them about 40 minutes to copy their DNA, but they will start copying their DNA. So once the DNA is copied here, okay, maybe at a point of origin right here, it'll start copying in both directions, and it'll start making new copies of the DNA before the cell even divides. So it starts making copies of the copies of the copies of the copies. And so the cell can divide every 20 minutes. Every 20 minutes it splits. So it has to actually start copying or replicating the DNA ahead of time to kind of stay ahead of that process. And that's why bacteria can grow very quickly on a surface or inside your body or under ideal conditions. In eukaryotes, okay, remember eukaryotes have a nucleus and other membrane-bound organelles. Okay, we're going to talk about division, cell division this chapter. We're going to focus on eukaryotes. It's going to happen in two steps. The first step, mitosis, is just the division of the DNA or what's in the nucleus. So when we go through these stages like prophase, metaphase, anaphase, telophase, we're going to be looking at these little X-shaped structures that look kind of like this. You can see the little red ones in the cell, and how they are divided and pulled apart so that the daughter cells here each get a copy, because it's very important that the cell makes copies and passes on that DNA to their offspring. The actual splitting of the cell, the pinching into two new daughter cells is called cytokinesis. Cyto meaning cell, kinesis meaning splitting. Okay, so that's the actual process. So eukaryotes, we'll get into more in future videos. Okay, one of the first things before we actually discuss division is this idea of a cell cycle, just the normal daily life or life pattern of a cell. Okay, so repeating series of events that include growth, replicating the DNA, and then actually dividing into two. So kind of the lifespan of a cell. We call it the cell cycle, and we give the stages names. The first one, G1, okay, you see down here, we used to call it gap phase, but now I like to refer to this as growth because you get a very intense growth. This is coming just after cell division, and you're now just a new little daughter cell. You're going to be soaking up nutrients and oxygen, and you're just going to be growing and growing and growing and building cell parts. And also, if you're part of the body somewhere or a plant or something like that, you'll have certain functions that you'll need to do as well. Okay, once it gets bigger, 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 and it says, I better start thinking about dividing. The next step over here is S phase. S stands for synthesis, or that is when DNA replication happens. Okay, DNA replication is when the DNA copies itself because it needs to have two copies of all the genes of the DNA to give to each of the two new daughter cells. Okay. Next stage is G2 or second gap. Here you've got more growth. The cell is also going to make proteins needed for cell division. So when we start talking about things like spindle fibers and stuff like that, this is made during the G2 stage because it's thinking, I'm getting bigger, bigger, bigger. I need to think about dividing. Okay, so some of that is done at G2. At the end of the G2 stage, these first three, I didn't learn when I was in high school back in the 90s. We just used to refer to these as interphase. And some scientists still do. You can see that on the outside, this darker green ring is still called interphase. Okay, now we know interphase was most of the life of the cell. Okay, now we take that and we split that up into smaller steps where we can recognize growth happening, DNA synthesis happening, and then even more growth and kind of making some of the structures that are needed for cell division. Okay, the M stage is the mitotic stage here in red. Okay, the mitotic stage is actually split into smaller phases, okay, which we will look at in the next video, the actual stages of division. Okay, that is mitosis, the division of the nucleus, and the actual division of the cell, which we call cytokinesis. Okay, so the cell divides, and then you've got two daughter cells. Now, one thing I need to note that I added in here is you'll see this resting or G0 stage that kind of loops back around in the G1 stage. Some cells, after they divide and they grow and they grow and they grow and they kind of determine their function, they become specialized. So we'll write in your notes here, we'll write cell specialization. Okay, some things like nerve cells, muscle cells, won't divide on a regular basis unless there's an injury or something like that, and some don't at all. Okay, we know that some parts of our cells, some parts of our body tissues that we may damage may not regrow. Okay, it might be permanent because they are stuck in a G0 stage and will not divide and make new cells. Now, during the life cycle of the cell, so here's that cell cycle figure again. Now you see some stuff added in there. Okay, there are what we call checkpoints, where there are regulatory proteins that the cell uses as signals, kind of like a traffic light: green light, yellow light, red light. Okay, green light means go. So after the cell divides, after the mitotic stage right here, the first checkpoint is called the cell growth checkpoint and happens after the end of G1 phase. And the cell is going to check itself. It's going to say, am I big enough? Do I have all the proteins I need for DNA synthesis? Am I ready for the next stage? If not, it's going to go back through the growth phase again. Okay, so it's just a helpful check to make sure the cell is on track and has the proper things it needs. The second one happens during the S phase. Okay, and here we're just going to make sure whether the DNA has been replicated or copied properly. Okay, if a mistake has been made, or it's not finished copying, or something is wrong, it's going to initiate a repair so that we don't have a mutation or something that we are going to pass on to cells that are going to keep passing it on. So that's a very good thing that our cells check the DNA and make sure everything is in order. The last one, the third checkpoint that we often talk about is during mitosis, during the M phase, during metaphase. During metaphase. In metaphase, all the chromosomes, those X-shaped structures, are all going to line up in the middle of the cell. And these spindle fibers are going to reach out and grab them and pull them each direction to the daughter cells. Well, it's going to pause here and check just to make sure that all those chromosomes lined up in the middle of the cell all have a spindle fiber attached to them and are ready to start pulling. We don't want to start pulling before all of the spindle fibers have taken hold, or you're going to get the wrong number of chromosomes divided up in the wrong daughter cells. And that's not good either. It's going to mess up our chromosome number and the amount of DNA and the genes that need to be divided equally. So that is a very important stage as well. And these checkpoints just help the cell to make sure everything is going properly. If not, it goes back and tries to fix it. If not, it goes through what we call apoptosis, kind of a programmed cell death, and it will sort of destroy itself so that it doesn't keep generating mistakes or problems in future generations of cells. Sometimes when we see those checkpoints mess up, kind of those green lights, red lights, or I know our AP book refers to them kind of as a gas pedal and a brake pedal in your car, whether to speed up the cell cycle or slow down the cell cycle. Sometimes those checkpoints get skipped. Sometimes they're overlooked. Sometimes people run red lights, and that can cause bad things. When the cell cycle is running out of control, that's called cancer. Cancer comes from our own body tissues when the cell cycle is no longer regulated by those checkpoints. And it just starts spinning out of control. The cells start dividing. And so where you normally get this kind of nice rows of normal cells in a tissue, now they're dividing like crazy, often enlarged. They don't look like their normal cells. And you get this big clump or mass of cells called a tumor. Okay, these tumors often start out benign or localized in a certain area. And as they grow, sometimes they can break through membranes. Sometimes you might have a blood vessel here where these things can metastasize or break off and lodge in other parts of the body, and it can become very serious. And that's why early detection is key. Cancer is really bad because you can see over here, this is somebody's liver that they did a cross-section of. I know, kind of gross, right? But it gives you an idea that here, this nice red tissue in here, that's normal liver cells functioning, trying to do what a liver cell needs to do. Okay, these big lighter colored blobs are big tumors of cells, cancerous cells. Now notice they're taking up space from the good normal functioning cells. They're also taking up nutrients. They often will cause blood vessels to grow more towards them to feed them more oxygen and nutrients. And that's really going to disrupt and affect the function of this liver. And eventually, when cancer, continues untreated, it can cause the shutdown or the death of that organ. And obviously, as vital organs start to get cancer and get shut down, and if it spreads to other organs, it can lead to the death of the organism, which many of us have been touched by cancer, either some way or shape or form within our families and family structures. And it can be really sad, and there's a lot of research going on how can we get to these cells and get rid of them, okay, so that they don't cause this damage? How do we detect them early, and how do we get rid of them? Okay, but again, it goes back to something in real life. It goes back to the cell cycle and how cells work, how biology works, how cells divide. Okay, so just kind of an overview of this chapter, or in this video. Next video, we want to look at the mitotic stage more specifically and look at each of those stages they go through.